right, well, good afternoon, everyone. I'm Randy Sommerfelt, and my project is Physicochemical and Respiration Dynamics of Mars soil. As most people would discuss about Mars, the one question that we always brought back to is, has Mars ever supports life, or does it support life now? And the best way to answer that is if we go there. However, due to complications, the best way to do this is to do some analog research beforehand. When most people start looking for life, is looking for water. However, an overlooked source is the soil. So my project was to collect desert soils and analyze it for the basic and physical, physical and chemical parameters to show the possibility of life. My analog, well, the most part, we all know why we go to MDRS. It's really closely related to the Mars environment. And well, my project would help going out there to MD, the Mars Desert Research Station, would be a valuable research to help develop techniques to collect sources if we would ever go to Mars. And my research goal was to identify 5 sites of different areas, such as one being wet and one being dry, or it's really bright in one area versus the shadowy area. For each site, I took a few soil cores, and then I ran it through some testing in the lab back in the half, just to see any microbial growth. And here are some of the testing parameters I took. I tested for pH level, water percentage, soil texture and composition, nitrogen, phosphorus, and potassium, and soil respiration, along with light and temperature. And along with my research, I came up with a few objectives, which was to learn the process of time to research and finding hypothesis testing, develop scientific questions and formulate ideas on how to answer these questions, and also to acquire the skills by operating these equipment out in the field and in simulations. And after which I critique and evaluate and synthesize data, and I pretty much catalog it together to come up with data and graphs to help convey this information. Of course, this project has also helped me become more well-balanced because I have a passion for science, but not yet a scientist. I'm still a student. Can't wait to go out there again in the field. And of course, it was fun and developed a deeper, richer role for science. And I grew close relationships to my crew, as you would if you were to go to manned missions. You need to be really close to your crew to be an effective team. Besides basic equipment such as writing utensils and paper to write down the data points, and a laptop to put all of this on an Excel sheet. Some of the equipment I used was a soil texture kit, a soil sampler, a pH meter, NPK, soil testing kit, soda line, and a camera. Also, I would use baggies to seal the soil after they're taking the sample from the sites. And I also would use jars and lids to simulate a closed environment, which I would use with a soda line. And so the procedures I used was wet weight versus dry weight. I would measure the weight of the soil, then I would put it through the oven at 105 degrees Celsius for about an hour just to get the dry weight out there and then factor that in. The soil respiration was the major part of my research, which it's actually a pretty simple procedure. It's you would use soda lime, in which as microbials digest and metabolize, they give off CO2 in the soda lime. Kind of evaporates away with it. And so with that, you can turn in respiration microbial activity. And then with the micronutrient testing, I determined the composition and NPK. For preparatory lab work before going out into the field, I would incubate the soda lime in the mason jars, since that would be tough enough to take the heat at 105 degrees Celsius for 24 hours. It's recommended for 48 hours. However, I only had a week to do this research, and so I just did 24 hours and factored that in later on. And I made a list what I need to take out when I was out on EDAs to what I need to do. And testing out in the field was basically, it actually was pretty hard on my knees and the equipment, but it was, as I developed techniques and how to do it, it was good to have all the equipment simulation to know if I could go man mission, kind of working. Anyways, take the soil sampler, get soil cores, throw it in a baggie, take it back to lab. But also, use there's a cake lid. Put the soil lime that's already in the jars, and I would unscrew the jars and quickly make a sealed environment with the cake lids. So test an area of where I took the soil cores out, then come back to later on, the next day after 24 hours, and bring it back to the lab. And where in the lab I would test out, go through testing parameters. Here, my research, due to resources, and my budget was about 300, so I had to do a lot of qualitative, but it was kind of quantitative research, so it was just low, medium, high, and you can see me here, I had mixed in the soil with what I need to determine how much nitrogen, potassium, phosphorus was in it. And as I was doing this acid, I would dry out the soda lime again once after taking a soil canvas in the oven for a while. So I was doing two things at once. And afterwards, I brought up that I had a catalog and looked at all these numbers for about a day before I figured out what would be the best way to represent it. And this is just basic stuff for the soil composition and nutrient contracts. And 16 moles of it is sand and clay, that's B for the Desiparium. And the interesting thing, everything was low in nitrogen, just like Mars would be. Everything was pretty high in potassium. But really cool stuff was comparing the soil respiration to the light and water percentage and temperature. And you can see some of these graphs as light intensity it correlates with respiration. The brighter was an area, the more respiration was getting off. So maybe photosynthetic microbial life was to be help with activity. There was really no correlation with water. The temperature was an inverse correlation. As it was colder, there was more respiration, so maybe the life there was more climates due to cold weather. And it's just kind of in words, you can see temperature, respiration has a correlation, but yeah, also saying nitrogen as well, also it's a factor essentially like biological molecules. And where it was determined that the soil respiration was the highest in the balanced sand and clay samples, where it was balanced. And when it's warm, it's soil sand, and in a desert, it's going to be sandy. But some more points being given off again. Well, levels of nitrogen resistance for the actual Mars surface. In further studies, if I was to have a bigger budget and had more time, I'd love to take a larger sample size and more of it at each site and do more sites also. Longer periods of sampling, incubate for 48 hours. Use a PCR kit to determine the major bacterial ataxia, and just kind of do comparative study if we were to ever get samples from Mars, compare the soil of Earth there, to see kind of the evolutionary process in microbiology, and to see if there's any organic matters in carbon. That's most of it as it is, and does anybody have any questions? Yes?
So what challenges were presented to you when you were in the super sampling as opposed to when you were sampling
It was kind of hard to take samples.
Please repeat the question.
The question was what kind of challenges was it to take samples in a suit compared to not in a suit? And well, some of it was getting how to handle the sampling tube and carefully not touch the sample as much as possible as it storing it and taking it back to cab safely. And we did get a little rain there, so that did affect some numbers in a couple sites, not all of them. So that factored in, and it's kind of hard to actually get some of the sample cores due to the composition of it. Any other questions? Yes.
Could you please go back to the slide where you've got the plots and the data on for a second?
What's it?
Could you please go back to the slide showing your data where you fitted the lines?
Oh, yes. Is this one?
And just, yeah, that's the one. How accurately measured are these data points? Because you fitted these, presumably you've used some kind of linear regression to fit these lines. I'm just wondering how statistically significant are these linear fits? Because that data does look quite noisy. So can you put a p-value on this?
Yes. Well, I don't really get any, like I said, no really correlation on there. And we had, there was, with the since it was qualitative research, we probably get numbers, but they had formulas with the kits I was using. So it took a while to actually get these graphs. I have most of my research notes on how to do that with me. And so if you would like to know, you can email me or my advisor, Dr. Christian at the Clinic Community College.
Thank you.
Yes, ma'am.
I'm interested in your graph of respiration versus temperature. Was that the cultivation temperature or the temperature at which you collected the microorganisms? What temperature was this?
Well, I collected temperature each day just to get an average in at the sites I took. And well, I was tested at Celsius and got a rough estimate. I had to convert it to Fahrenheit to get better representation and a better number.
Those temperatures are the temperatures of your soils.
Yes, they are. And the area of the soil, the area of where I took the size of the soil samples. Yes.
And in the soil.
Yes, I was sticking the temperature and temperature measure, which was also worked as the light intensity and all that stuff was 3:1. And I was sticking into the soil to get a rough estimate. I took several estimates just to get a good number.
How come you came to no conclusions on your respiration versus percent water?
Well, the numbers I got was just iffy, just like I said. We got some little rains, so that affected a few of the sites that I had measured. So it really was inconclusive data to use. So if I was to go back there again, I would definitely use more data points with it.
Okay, but what about the ecology?
Ecology?
Yeah.
Not
I'm
sure.
sorry, I'm in teacher mode making.
No, that's fine. I hopefully
Right.
get to create each and every.
But you missed a valid point that you should have made in the conclusions.
Okay.
Or under percent water. And it's fine if you make it. Just think about it.
Oh, definitely.
And this is the first project that I ever did at MPRS. So, and it was my soil line up there. And we have tons and tons of data. So if you ever want extra data to look at, I have years worth of data.
Oh, that would be greatly appreciated.
So,
All right, thank you. Yes, sir.
Yeah, where is McMillan Community College?
It's in Waco, Texas. Yes, sir.
So can your experiment compare with Weidenländer labeled release?
Hmm?
Can your experiment compare with the Weidenländer labeled release light detection experiment?
Compared to what? I'm sorry.
Weidenländer light detection labeled release experiment in 1976.
Okay, you might have to ask me that after. Okay, thank you. Any more questions?
How deep were your soil samples? And also, did you take into account runoff with rain? Where were they located?
Yes. Well, I didn't expect when I took two of the sites got a little bit, but one of them was really kind of a bit moist, but it wasn't too much that I couldn't use data for. But took the soil samples, the soil cores were 6 inches deep in the soil, so get a good temperature reading and all that. And yes, I did take into account to the water and didn't expect it to, right, because there was only one that I really didn't expect to get water. It was right next to that, about 100 feet off the path, where it was always getting bright intensity from the sun. All right, well, thank you. If you have any more questions, see me afterwards.